When a wound refuses to heal, the answer lies hidden in its pus. Today, let's uncover the world of pus culture and sensitivity. Pus culture is one of the most important diagnostic tools used to identify the bacteria responsible for wound infections, abscess, and deep tissue infections. By isolating the organism and performing antibiotic sensitivity testing, the clinicians choose the most effective treatment and prevents the complication. Let's see the types of sample under pus culture. The common samples include aspirated pus, which are the most preferred sample and are collected using sterile syringe or needle directly from the abscess. The second category is swab samples. They might be single or double swab from infected wounds, ulcers. These are least ideal, but they are the commonly received. Next type is drain fluids, which are collected from surgical drains or wound drains. And the last category are tissue bits. Let's dive into the processing method of pus culture. The first step is sample reception and labeling. Check the patient details, type of sample, and the time of collection, followed by direct examination. The process of gram stain, which shoot out the pus cell and the organism present in the sample. A clear morphology test should be done, which gives a early clue to infection. The important step of the pus culture processing is inoculation or streaking. Once the pus sample reaches the lab, the most important step is proper inoculation. This ensures that we isolate the causative organism clearly and avoid mixed growth. The choosing of right media for pus culture is crucial. For pus samples, we commonly use blood agar, McConkey agar, and chocolate agar. Blood agar, which helps in the growth of most pathogens and observe the hemolysis. In case of McConkey agar, they helps in differentiating lactose fermenters and non-fermenters. The chocolate agar is commonly used for fastidious organisms. If you have a double swab for processing, the first swab can be used for smear preparation and the second swab for culture. For culture, hold the swab gently like a pencil. Touch the swab to the surface of the agar. Roll and rotate the swab in the primary area to release maximum organisms. This is called the inoculation zone. The streaking technique. Use a sterile loop to drag the inoculum from the primary area into the secondary areas of the plate. Use the 4-quadrant streak method: primary, secondary, tertiary, and quaternary. In case of primary, the heavy inoculation zone is seen. Secondary, the organisms are diluted. In tertiary, the further dilution takes place. And in quaternary, the isolated colonies can be seen. The common wordings heard in the microbiology lab are proper streaking is important. And why? Because separate mixed bacteria into isolated colonies. They prevent crowding, it identifies the colony morphology, and improve the accuracy of antibiotic sensitivity technique. After the inoculation process, the next process is incubation. The plates are incubated at 37 degrees Celsius for 24 hours. The chocolate agar in carbon dioxide atmosphere if required. On the next day, based on the colony morphology, gram stain, and the biochemical test are performed. Perform the antibiotic sensitivity test manually or by using automated mechanism. And report sensitive, intermediate, and resistant patterns. The general precautions that have to be kept in mind before performing pus culture. Collect the sample before antibiotics whenever possible. Use sterile containers for sample collection. Transport the sample to the lab quickly. Follow biosafety precautions like gloves, lab coat, and proper disposal. Use laminar airflow for processing. Every sample we process brings a patient one step closer to healing. This is Sneha signing off, and I'll catch you again in the next video only on Medical Guruji. Until then, stay safe and bye-bye.